Hey class, let's focus in depth now on the second line of defense of our immune system. The second line of defense is the interface between our generalized first line of defense and our more specialized adaptive third line of defense. So we're going to have multiple immune responses interacting with each other. Some of these specific immune responses and general immune responses will have specific interactions that can cause phagocytosis or inflammation, fever, and can involve specific antimicrobial proteins in our bodies. So let's talk about phagocytosis. This process of phagocytosis is the cornerstone. It is the linchpin that links our first and third lines of defense together. As we look at phagocytosis, we're generally speaking going to have a white blood cell identify a pathogen that does not belong in our body and then engulf and destroy that pathogen that does not belong in our body. The white blood cells are going to be surveying the tissues of our body. They'll move through these compartments and remove anything that doesn't belong. These molecules or cells that don't belong in our body are going to have specific immune system information removed from it. So as a bacterium is phagocytized, that bacterium is going to be pulled apart, and then specific molecules from the bacterium are going to be harvested and used to initiate the immune response, or the specific immune response. Our three primary phagocytes are our neutrophils, monocytes, and macrophages. The neutrophils are a general purpose phagocyte. They are the most common white blood cell in our body, and they are also the first responders. They are the first to arrive at the site of infection or damaged tissue. Because they're the first to arrive and they're at the highest concentration in our body, they are typically going to be the primary component of pus. So think of the whitehead, or think of that white oozy substance that results from a wound that's infected. That's going to be mostly dead neutrophils. We also have monocytes and macrophages. These are rarer than the neutrophils, but they are more accomplished when it comes to phagocytizing pathogens. Monocytes are transformed into macrophages. A monocyte's a very large one nucleide or uninucleide white blood cell that can be activated and turned into a macrophage. And as it's turned into a macrophage, it can leave the bloodstream and travel through tissues to phagocytize pathogens. A key difference between a monocyte and a neutrophil is the neutrophils will kill themselves in the process of destroying other pathogens. Monocytes do not kill themselves. They aren't kamikaze cells, so to speak. We also have histiocytes. A histiocyte is a specific kind of macrophage that's localized to a specific tissue. Histio, or hist, is the word for tissue. Think of histology, the study of tissues. A histiocyte is a tissue-specific macrocyte. So if we look at our lungs, we have macrophages, or phagocytes, that are specific to the lung tissues. We have Knupfer cells that are specific to the liver, dendritic cells specific in the skin, and we'll have other varieties of macrophages that are localized in the spleen, lymph node, or other tissues of our bodies. Here we can see an electron micrograph of a lung alveolar macrophage. Here in the liver, we can see a diagram of a Knupfer cell. And then here in the skin, we can see a diagram showing the dendritic cells. These are macrophages, or phagocytes, that are specific to tissues of our bodies. In case you didn't know, or remember from the prior video lectures, a phagocyte is literally an eating cell. Phago means to eat, cyte stands for cell. Phagocytosis is the process of bringing a solid substance into the cell. So it's when the cell will physically engulf that solid substance. Sometimes it's a molecule, or sometimes it can be an entire cell. During phagocytosis, we'll first have chemotaxis, followed by ingestion, then phagolysosome formation. Finally, it's destruction of whatever was phagocytized, and then excretion of the byproducts. And we'll talk about these events in more detail. First, to initiate phagocytosis, our phagocytes need to recognize specific markers on the cells that need to be destroyed or phagocytized. Those are going to be known as PAMPs, pathogen-associated molecular patterns. These PAMPs signal the phagocytes to phagocytize something that doesn't belong in our body. They are shared, these molecules that are going to be located on the surface of the cells and are present on many different organisms, but not present in mammals. So this is a way that our body can tell a foreign intruder apart from something that does belong in our bodies. These are the red flags, so to speak. A bacterial PAMP can include peptidoglycan. Only bacterial cell walls have peptidoglycan. We do not have any peptidoglycan present on any of the cells of our body. So if our white blood cells, our phagocytes, find peptidoglycan, that's something that can be used to say, hey, this is something that doesn't belong here. It's a big red flag. Another one is lipopolysaccharide. If we're working with our gram-negative bacteria, which will have the lipopolysaccharide represented, or if we have a gram-positive bacterium, there'll be peptidoglycan on the surface of its cell envelope. Something that's specific to only viruses is double-stranded RNA molecules. As humans, we have no double-stranded RNA. So if we ever find double-stranded RNA, that's a big red flag that something is amok, something is not right in our bodies, and we should phagocytize it. Our phagocytes will use PRRs, pattern recognition receptors, to recognize our PAMPs, our PAMPs. These are receptors found on the surface of our phagocytes. They are going to be able to bind to the PAMPs, and as we have a PAMP bind to a PRR, the cell will initiate phagocytosis. An inflammasome is a special structure within a phagocyte. The pattern recognition receptors found in the cytoplasm of a phagocytotic cell can initiate or trigger our immune responses. If we recognize a PAMP with our PRRs, once it's been phagocytized, we can have chemical signals released from our phagocytotic cell. These chemical signals that are released are called inflammasomes and regulate the inflammatory response. So if we look at this process, we'll first have chemotaxis, the first step of phagocytosis. We need to have our phagocytotically active cell move towards the pathogen that doesn't belong. Generally speaking, there's going to be a chemical gradient attracting it. Then we'll have adhesion occur. During adhesion, our bacterial cells will have their PAMPs bind to the pattern recognition receptors on the surface of a phagocytotically active cell. Those PAMPs and PRRs are then going to initiate engulfment, where there's an invagination of the cell plasma membrane of our phagocyte. A phagosome then forms. A phagosome is a small lipid-bound vesicle that contains whatever has been phagocytized. The phagosome, generally speaking, is going to fuse with lysosomes. And the lysosomes containing digestive enzymes fuse with the phagosome and release their digestive enzymes into the phagosome. Those digestive enzymes then will destroy what is ever in the phagosome. There'll be many highly oxidative free radicals present from the lysosomes that will destroy and break apart the bacteria and or viruses that make it in, generally speaking, bacteria that are present in our phagosome. And then at the end, the cellular debris that is left over is released, and specific parts of our destroyed or phagocytized bacterium are going to be saved and used to initiate that immune response for an acquired immune system. So to overview or summarize the slide, first we have chemotaxis. We attract the phagocyte to the bacteria. We have adhesion. They bind to each other, and then the bacterium is swallowed, and then a phagosome will form around the bacterium. The phagosome will then bind with a lysosome, forming a phagolysosome. The bacteria is then destroyed, and then the leftovers are eliminated through exocytosis. During the inflammatory response, we're going to have a very complex series of reactions occurring in concert with each other. We're going to have a very classic series of symptoms associated with inflammation, one of which is rubor. Rubor is a fancy term that indicates that there's redness, and that redness occurs from increased circulation and vasodilation of the injured tissues. As there's increased circulation, there will be more heat delivered to the tissue from the body's core, which I was going to call calor, and that inflamed tissue will be warm in addition to red. The increased vasodilation, generally speaking, is also going to associated with tumor. So there'll be liquid leaving the blood vessels as the blood vessels become vasodilated, and that liquid causes swelling, also known as tumor. So rubor means redness, calor means warmth, tumor means swelling, and then dolor is a technical term that refers to the pain as the naked nerve endings are stimulated. And if there's too much inflammation, there can be a loss of function of that tissue. Depending on the disease, there may be chronic inflammation caused by that disease. This chronic inflammation can be localized in the body, or it can be system-wide or systemic in our body. Aging has been associated with inflammation in several research studies. And if we minimize that inflammatory response, it's been found that we can minimize the effects of aging. These studies have been primarily done in organisms that have short lifespans, such as lab mice and lab rats. Getting good data on human beings is a little bit more difficult because the studies will take decades to complete. Some of the things that are going to cause inflammation to occur in a human host include trauma from infection, or we can have direct tissue injury. Sometimes necrosis can occur either from a direct physical assault to the tissue, so think of a crush injury, or we can have a chemical agent. So let's say the patient has drain cleaner exposed to their hand for an extended period of time. Or we can have a specific immune response triggering the inflammatory response. Why do we have inflammation? It's got to do something good for us, right? It can't all be bad. Depending on the degree of inflammation, we can use inflammation to mobilize or attract other components to the site of injury. Think of the vasodilation. As the blood vessels dilate, we can have more white blood cells potentially travel to the site of infection. And as those blood vessels become increasingly leaky, it's easier for white blood cells to leave the cardiovascular system and get into the extracellular fluid. Furthermore, as we have this increased vasodilation and increased blood flow, we can remove damaged and harmful tissues and harmful substances immediately for the short term. And as this vasodilation occurs and there's the tumor setting in, we actually are going to slightly pinch off the blood vessels so that after that initial response where we can flush away components from the site of inflammation, after a short period of time, there's actually a decrease in blood flow that makes it more difficult for microbes to enter the bloodstream and be circulated throughout the rest of the body. So our inflammatory response is a very powerful defensive reaction. It's not all bad. The bad part of our defensive or inflammatory response is when it is going out of control, when there's too much inflammation, and that tumor completely pinches off the blood vessels. That is not a good thing. We want a mild constriction of the blood vessels from the tumor, not a complete pinching off of the blood vessels from the tumor. Inflammation will allow our body to maintain a stable homeostatic environment and speed the response as we heal ourselves from an injury. Inflammatory response, though, can cause tissue injury if it's overstimulated. So if we look at specific microbes, some of these specific microbes are actually going to cause damage to our body by eliciting excess inflammation and then have additional cells that they can use for a source of nutrition. One of the specific kinds of chemicals that mediates the inflammatory response is known as a cytokine. A cytokine is a generic class of chemicals that is going to regulate that inflammatory response and signal the chemotaxis of our phagocytes. Our cytokines can be produced by a wide variety of cells, including our monocytes, macrophages, lymphocytes, fibroblasts, mast cells, platelets, and endothelial cells. So lots of cells can make cytokines. Depending on which cell type and the number of cell types are producing cytokines, the effect of inflammation can be localized, or it can be short-term, it can be long-lasting. It all depends on the concentration and the duration of cytokine production. So we can have different kinds of cytokines within our broad category. These different subcategories of cytokines can mediate nonspecific immune responses, such as inflammation or attract those phagocytes to stimulate phagocytosis. Some cytokines that activate immune reactions during inflammation are going to have vasoactive mediators. So they are going to cause vasodilation to initiate the inflammatory response. Other cytokines can regulate the growth and activation of T and B lymphocytes, and some cytokines are going to initiate hematopoiesis, the production of blood cells, specifically for white blood cells. And there are a wide variety of other cytokines that we aren't going to talk about in specific detail. So if we look at our cytokines, you can see that there's a long list on this table with specific kinds of cytokines such as interleukin-1, interferon gamma, bradykinin, histamine, prostaglandins, leukotrienes. I do not want you to focus on the specific kinds of cytokines. I want you to focus on these big headers. We have cytokines that can initiate inflammation. We have cytokines that activate immune reactions. We have cytokines that are going to be vasoactive. They initiate vasodilation. Or we can have cytokines that cause lymphocyte development. And then our miscellaneous categories as well. So focus on the category headings, not on the specific kinds of cytokines. During inflammation, we're going to have this series of events occur in a predictable and coordinated fashion. The first thing that we need to have for inflammation to occur is the initial injury. And once the initial injury takes place, the chain reaction of events of inflammation will be initiated. So first, we have the injury occur. And during that puncture, that injury, that physical object can inject or insert bacteria past the epithelial barrier into our bodies. The initial response is going to involve changes to the blood vessels. We're going to have vasodilation, and that vasodilation is going to allow for phagocytotically active cells to exit the bloodstream and get to the bacterial cells in question. We are then going to have more and more white blood cells, particularly neutrophils, enter the site of infection to destroy the bacterial cells that are rapidly reproducing. As more and more neutrophils enter the site of infection, they will die off and form pus. After we have destroyed the bacterial infection, we are going to have additional white blood cells migrate to the site of infection to remove any straggling or remaining bacteria, and then we will repair the damage to the tissue. So we have step one, puncture. To initiate the inflammatory response, we have mast cells that release cytokinins that initiate the inflammatory response and attract our phagocytes to the site of infection. The first phagocyte to the site of infection will be our neutrophil, and then we'll have macrophages and or lymphocytes as the final phagocytes at the site of infection. The specific name that refers to the movement of a white blood cell out of the bloodstream is referred to as diapedesis. Di is a prefix meaning two. Ped is a word meaning foot. It looks like a two-footed cell as it's squeezing out of the blood capillaries. So diapedesis is the movement of a white blood cell out of the blood capillaries into the body's tissues. The endothelial cells that line our veins are going to have adhesive receptors that can capture the white blood cell and get them to exit the bloodstream. These adhesive receptors are activated in response to cytokine exposure. After our white blood cells exit the bloodstream, they're going to move through our body's tissues, being powered from a process known as chemotaxis. During chemotaxis, the white blood cells will move from an area of low concentration of the stimulatory chemical to an area of high concentration of the stimulatory chemical. That chemical that powers chemotaxis, generally speaking, is referred to as a cytokine. Recall, one of the functions of cytokines is to attract our phagocytotically active cells. So diapedesis and chemotaxis work hand in hand with each other, work in conjunction with each other to get a white blood cell from the bloodstream, out of the bloodstream, and to the site of infection. So here we can see a neutrophil. That neutrophil is going to find a gap in a vasodilated blood cell. It will exit the blood cell via diapedesis, and once it exits the blood cell and is in an extracellular environment, cytokines or other chemical factors will guide the white blood cell to the site of infection as the white blood cell seeks out areas of higher and higher concentrations of the stimulatory chemicals. Some of the benefits of edema and leaky vessels include an influx of fluids to the extracellular environment. This influx of fluids during the inflammatory response helps to locally dilute toxic chemicals that may cause cell death. We're also going to have a fibrin clot form, and this fibrin clot is a specific mesh of proteins that helps to keep microbes from spreading further throughout the body. We'll have lots of neutrophils get to the site of infection and exit the blood cells or blood vessels easily because the blood vessels are so leaky. As I mentioned before, neutrophils make up the majority of pus. Pus is that whitest, liquidy-ish tissue that is made of liquefied bacterial and cellular debris from our bodies. We're also going to have pyrogenic responses occurring. A pyrogenic response is a response that is going to stimulate the formation of pus. This response can occur from many different kinds of bacteria that are typically located on the surface of our skin. So our streptococci and our staphylococci are typically associated as skin bacterium. We can have our gonococci, think of gonorrhea and a pussy excretion from the urethra. Or we can have meningococci, which can cause a pussy buildup of the meninges of our bodies. Side by side with infections, we'll typically have a fever occurring in our body or a slight elevation of our body temperature. This abnormal elevation of our body temperature is going to be above and beyond the average body temp for most people of 98.6 degrees Fahrenheit. It's almost universally associated with all infections. The fever can be temporarily lowered in response to specific medications, such as aspirin or other prostaglandin inhibitors. But generally speaking, if there's no specific medication inhibiting the fever formation, an infection will always be associated with a fever. Many times we have fevers of unknown origins in our bodies. These are going to be intermediate fevers where our body temp is greater than 101 degrees Fahrenheit. These fevers are typically going to last for longer than 3 weeks for no known cause and can be determined or classified as a fever of unknown origin if there's been 1 week of inpatient investigation without a resolution, without a specific cause being identified. Obviously, if there's a fever of unknown origin, the physician is going to take a generalist approach towards treating the fever. But if the physician doesn't have a specific diagnosis, physician will take a conservative approach generally and give broad treatments for the fever. Our typical body temperature is maintained at 98.6 degrees Fahrenheit, but many healthcare organizations, thankfully, are moving towards the metric system. So think 37 degrees Celsius. 37 degrees Celsius is average body temp. A low-grade fever is between 37.7 and 38.3 degrees Celsius. Anything that is over 40 degrees Celsius is a high-grade fever and a big deal. It's very concerning if your body temp reaches that high. Chemicals that stimulate an increase in body temperature are known as pyrogens. An emphasis on that R right there for the pyrogen. Pyrogens have at the root word pyro, or heat, fire. So a pyrogenic chemical is a chemical that stimulates or increase in body temperature. We have exogenous and endogenous pyrogens. Exogenous pyrogens are going to be excreted from the pathogen and can be injected. Endogenous pyrogens are going to be removed from the pathogen after phagocytosis, and it's going to be produced by our bodies to increase our body temperature. So the exogenous pyrogens are in response to the pathogen acting on us. The endogenous pyrogen is going to be excreted in our body's response to phagocytizing the pathogen. And fever is not all bad. Mild fever is actually pretty good for us. Mild fever will inhibit the multiplication of temperature-sensitive microbes. Most temperature-sensitive microbes are going to multiply very well at 37 degrees Celsius, but at 38, 39 degrees Celsius, they don't multiply as well. A fever also makes iron less available in our bodies. As our body temperature increases, the production and activity of proteins that sequester iron in our bodies increases, making it more difficult for microbes to access that iron. And finally, an increase in body temperature is going to increase our metabolism slightly and make our immune system function at a more optimal and increased rate. So the production of new blood cells will be increased, the swallowing and destruction of pathogens is increased, and the diffusion of chemical mediators throughout the liquids in our body is also increased with that slight increase in body temperature. So the benefit of a fever can make a difficult decision whether to treat the fever or not. If there's a mild temperature, let's say of 99 to 100 degrees Fahrenheit, generally speaking, the approach is to sweat it out. But if the person is experiencing a high fever, anything at 100 degrees Fahrenheit or higher, at that point, the temperature is becoming so high that your body's own proteins start to denature, and neural function can start to cease. So if there's an elevated fever, that should be treated. Some side effects of moderate fever include an increased heart rate or an increased rate of breathing. So there could be tachycardia or tachypnea. Those are the technical terms. We'll also have a lowering of the seizure threshold. It's more difficult to have someone have a seizure induced if they have a mild increase in their body's temperature. Higher prolonged fevers, though, are bad for us and should be treated immediately. We also have in our body some antimicrobial proteins. These are small proteins in our body that are produced naturally by our white blood cells and by our tissue cells and are used to combat bacterial infections. These antimicrobial proteins originally were thought to be directed against viruses, but now we've found that they are involved against a wide variety of microbes, including bacteria and fungal cells, and to help regulate our immune system and to communicate between parts of our immune system. Some of these specific antimicrobial proteins include interferon alpha and beta and interferon gamma. So alpha was discovered first, then beta, then gamma. Think of the Greek alphabet: alpha, beta, gamma, delta, kappa, so on and so forth. So interferon alpha and gamma are going to be made by our lymphocytes, fibroblasts, and macrophages, and interferon gamma are produced by our T cells. These macrophages are part of our secondary line of defense, and the T cells are part of our tertiary or third line of defense. So by having these interferons, we can communicate between our second and third lines of defense and link them together. So what do these different interferons do? These interferons can bind to the surface of the cells and cause cells to change their genetic expression. Depending on what the interferon is, we can have a slightly different response, but regardless of what interferon we're looking at, they're all going to inhibit the expression of onconogenes, or cancerous genes, and suppress the effects of tumors. Some interferons are going to stimulate phagocytosis, and some interferons are going to help to activate the production of T or B cells. Now, for the test, I do not want you to focus on interferon alpha or gamma or beta and differentiating them. I want you to focus broadly on what interferons do in our body. So don't spend a lot of time differentiating or delineating between alpha, beta, and gamma interferons. Just know generally interferons are going to be chemicals that are secreted that stimulate phagocytosis or are going to help to regulate the activation of T or B cells or suppress cancerous cells. If a virus gets into a cell and infects the cell, our virus is going to ultimately destroy the infected cell. But that infected host cell can produce an interferon signaling molecule which will travel outside of its body and alert our immune system to the fact that it's infected. These interferon molecules will bind to nearby cells that are in the tissue, and those nearby cells are going to have the production of proteins that help make those nearby cells immune to the specific virus. If we have cell A, so we'll have a cell infected with a virus, that cell will emit interferons that travel next door to the adjacent cell and help that adjacent cell B be able to degrade and destroy any viral RNA that gets into it. And if we destroy the viral RNA, then we're not going to be able to translate any viral proteins in the adjacent cells. These interferons are not microbe specific and provide us with a valuable treatment for viral-based infections. Several antiviral medications mimic this category of interferons. And if we can stimulate our cells to destroy viral RNA, we can minimize the rate and the extent of a virus spreading through our body. So as I poorly drew on the slide previous, they can see a virus getting into the first cell. That first cell is going to have viral nucleotides in it, and that first cell will start secreting interferon molecules that can travel to the nearby adjacent cell, and that nearby adjacent cell, in response to those interferon molecules, will destroy any viral RNA that gets into it, preventing the translation and transcription of viral proteins. So we have interferons. To review briefly from interferons, interferons help to mediate phagocytosis, interferons help to activate T and B lymphocytes, and interferons help to signal nearby cells to become resistant to a viral infection. Let's move on to another category of antimicrobial proteins, the complement proteins. These complement proteins are called that because they can complement the immune system responses. And it's a category of up to 26 different proteins that work together to destroy bacterial cells and a couple of viral particles. It has a cascade reaction where we have a sequence of events that need to occur in order to activate our complement system. And there are three initial pathways we can use to trigger the complement system. But regardless of the pathway we use, they all are going to end up with the same response at the end. So to initiate, we're going to have our first complement protein bind to a foreign cell. This first complement protein is called C1. After our first complement protein binds, it's going to cause adjacent complement proteins to be chopped up and bind to the cell membrane of our bacterium that's infecting our body. After those adjacent complement proteins are attached, they are going to polymerize. They are going to start to assemble our membrane attack complex, the MAC. And this membrane attacks complex will initiate the next stage known as membrane attack. This membrane attack complex is going to form many small holes in the bacterial cell, leading to an equilibrium of the intracellular fluid and extracellular fluid in our bacterium. In other words, we perforate the cell wall and cell membrane of the bacteria, causing the bacteria to lyse. So we have three different ways to activate our complement cascade. We have the classical pathway, which is going to involve a foreign cell membrane of a parasite or surface antibody being bound by C1. We have an alternative pathway when the components of our complement pathway can be bind to specific pathogen membranes. This alternative pathway is quicker than our classical pathway. And then finally, we have the lectin-based pathway. We'll have mannose-binding proteins, aka lectins, that are going to bind to mannose, a specific kind of sugar residue that's on the surface of our pathogenic organisms and can initiate the complement cascade. Regardless of which complement cascade we initiate, they all end with the membrane attack complexes perforating the bacterial cell and destruction of our bacterial cell. So we'll have multiple proteins in cascade, and I'm not going to test you on which specific proteins do what. So don't spend time focusing on C1 or C5 or C9. What I want you to focus on is that there are multiple proteins involved, and these multiple proteins work together to make small perforations or holes in the bacterial cell wall and cell envelope, and that causes the bacterial cell to swell and eventually lyse. We also have antimicrobial peptides in our bodies. These are small chains of amino acids, and when I say small, I mean between 15 and 20 amino acids that can insert themselves directly into prokaryotic membranes. These are sometimes referred to as defensins or magnins or protegens. These are different kinds of our antimicrobial proteins. And they are going to have effects on both our nonspecific and specific immune system, particularly the second and third lines of defense. Right now, a hot area of antimicrobial research is finding a way to synthesize and utilize antimicrobial proteins to treat specific bacterial and protozoal infections in our body. Our antimicrobial proteins are going to be attracted to the lipids in a bacterial cell membrane. We'll have a positively charged portion of the protein that will bind to the phosphate head. We'll have a hydrophobic region that will penetrate the membrane. And as these antimicrobial peptides puncture the cell membrane of the bacterium, it'll initiate cell lysis and destroy the bacterium. But we need high concentrations of these proteins to kill a bacterium. So, concept check class. What are the steps of phagocytosis? Put them in the correct order. You can rewind the video, check your notes, or check your textbook. Go ahead and pause it and get me the answer. 1, 2, 3, 4, 5. So our steps are engulfment, killing and destruction, phagosome formation, adhesion, release of residual debris, chemotaxis by the phagocyte, and phagolysosome formation. We know that the release of the residual debris is the very last step. It has to be. There was a figure that I covered from your text that shows the release of the leftovers at the end. So by the process of elimination, the only option we have with number 5 at the end is option D. So we'll first start by our phagocyte moving towards the bacterium, step 6. Then we'll have the bacterium bind to the outside of the phagocyte. The PAMP will be recognized by the PRR. And then the bacterium is engulfed. After engulfment, it's going to be stored in a phagosome. That phagosome binds with lysosomes to form a phagolysosome. After phagolysosome formation, the digestive enzymes within the lysosome destroy the bacterial cell, killing and destroying it for step number 2. And then finally, the leftovers are released via exocytosis. If you have any questions, please feel free to post them on the discussion board or shoot me an email. Happy studies!